Okay, thank you, Alex. Right, so I'll start the first part of the presentation, and then Ashley, graduate student at Montana State with Ryan Thum, will be wrapping up the presentation. We just want to sort of acknowledge some other co-authors, important participants. John Jarrettson did a lot of the challenge test work that we're going to be talking about. Ryan Thum is sort of the lead geneticist and sort of doing all of our genetic analysis and providing lots of advice and input on this. And Alex, who ended up working with Ashley on the app that she's going to demonstrate for us today. Eurasian water milfoil was introduced in North America from Eurasia in the 1940s. It first showed up in Minnesota and Lake Minnetonka in 1987. It's now present in 48 states, and it's probably one of the most widely managed aquatic weeds in the US, certainly in North America, and possibly worldwide. It forms these extensive surface mats that shade the bottom, can outcompete native plants, and it also inhibits boating and swimming and other recreational activities. Now, there's a native northern water milfoil that's historically been in Minnesota. It's widespread. Prior to the introduction of Eurasian water milfoil, it was probably the most managed aquatic plant. And once Eurasian water milfoil was introduced, in some cases, it's actually outcompeted the northern water milfoil, especially in deeper water or poorer clarity conditions. So actually, northern water milfoil now has a little bit less, more restricted distribution than it used to have prior to the introduction of Eurasian water milfoil. These are both closely related. Actually, there was a while in the 50s when they thought that they were, actually one was a subspecies of the other in the same species group. They both vegetatively or clonally reproduce. That's sort of the most common way of them reproducing and spreading, but they can also reproduce sexually. Because they're closely related, they can interbreed and they form hybrids. And that's a hybrid between the invasive Myriophyllum spicatum and the native Myriophyllum sibericum. Now, these hybrids also reproduce vegetatively, but they can reproduce sexually, and that's important, particularly with the hybrids do seem to reproduce sexually a fair bit. So hybrid water milfoil was first identified, documented in Minnesota in the late 1990s, and sort of one of the areas that Michael Moody first identified. This hybrid water milfoil appears to be now fairly widespread across the northern states, from the mid-Atlantic to the Midwest and all the way into the western states. Most of the research that's been done on hybrid water milfoil has really been in Michigan, and then also expanding to Wisconsin, and for the past 5 or 8 years in Minnesota now. So why worry about hybrid water milfoil? Well, it's a hybrid, so it could be more vigorous or invasive, may show faster growth or be a better competitor. And certainly some work with Wes Gleason and Dan Larkin has shown that, and work also in Ryan's lab has shown faster growth rates and potential for competition in the hybrids. More importantly, the hybrids could be resistant to or tolerant of herbicides or biological controls. So we did some work with biological controls, and it really doesn't seem that there's a big difference between those. The hybrid is sort of intermediate in response to biological control agents to the northern Eurasian. And we haven't identified any real problems yet that hybrids might pose, but there's possibility that we could come up with new genetic variants. There is evidence for increased tolerance or resistance to herbicide in some populations. So there have already been populations, particularly in Michigan, that have been identified as being resistant to 2,4-D. Populations resistant to triclopyr, and then also different populations or genotypes resistant to fluoridone. So we could be, by using these herbicides, selecting for resistance or tolerant genotypes. And it may end up being more difficult to control these plants and require higher doses to end up getting control. So where is hybrid water milfoil and Eurasian water milfoil? Well, the DNR records accounting distribution of milfoil doesn't distinguish between hybrid and Eurasian. So they're both, originally all this was based on visual IDs. There are some genetic ITS IDs on those, but the distribution sort of includes non-native milfoil, which would be hybrid or Eurasian water milfoil. So currently, we've got these in about 400 water bodies. So that's either Eurasian or hybrid water milfoil. To figure out what you've got, then you need to do genetic analysis. So We ended up a number of years, probably like 2017, with funding from MACERC, started assessing lakes for a genetic composition. So now we've analyzed over 100 lakes in Minnesota using genetic markers, either ITS or now primarily microsatellites. And we've identified 68 lakes that have Eurasian water milfoil, 50 lakes that have hybrid water milfoil, and 36 with northern water milfoil. Now, interestingly, 43 of these lakes only have one taxon, but 46, about half of them, have two or more taxa. So they may have Eurasian and hybrid, or Eurasian and northern, northern and hybrid, and so on with that. The other thing that became clear from our work is, in general, northern water milfoil is more common in the outstate areas. So that's the blue dots here, and not as common in the metro. Hybrid water milfoil seems to be a bit more common in the metro. That's in these green dots. And Eurasian water milfoil in the yellow dots is fairly widespread, which makes a little bit of sense on that. Well, that's the taxon identification, but really, you really need to drill down and say, what is the genotype we're dealing with? So we're sort of increasingly realizing that you can't say, look at resistance or competitive ability just based on whether it's hybrid or Eurasian, but you really need to know what genotype or what strain it is. So Ryan's lab's been identifying water milfoil genotypes based on 8 microsatellite loci, and we've collected multiple samples per lake. So we've got a fairly good characterization in these lakes, often 20 samples per lake. So in Minnesota now, we've identified 17 genotypes of the Eurasian water milfoil. One of these is widespread. So it's found in 62 lakes. It's the one that's sort of everywhere. We did find 7 lakes that had more than one genotype, although generally most lakes only have one genotype. There is one genotype that's sort of other genotype that's sort of been found in 6 lakes. Otherwise, these other minor genotypes are restricted to 1 lake each. For northern water milfoil, we've now identified 96 genotypes, so more diversity in native northern water milfoil. And every lake we've looked at has a different genotype. And many lakes have multiple genotypes. Sort of the take home from this is northern water milfoil has been reproducing sexually. And so there's diversity within those lakes, and it's not spreading via boaters or whatever to other lakes. So it stays in a single lake that's there. Hybrid water milfoil is intermediate. We've got 89 genotypes we have identified now, and most lakes have one unique genotype that's not found in any other lakes. But we do have 17 lakes that have multiple genotypes. One lake we actually found 13 genotypes in. Be interesting to see if those persist. And we have a few genotypes that are found in multiple lakes, 8 genotypes so far that we identify. One of them has been found in 13 lakes, and then 7 of them are just limited to a minor number of lakes, 2 to 4 lakes. So this is the distribution of the widespread Eurasian genotype. Sorry about the map here. But there may be some lakes where, again, it's in with northern and hybrid, but all of these dots here are the widespread Eurasian genotype. It was probably the genotype that was initially introduced to Lake Minnetonka in Minnesota and is clonally spread throughout the state. And that's its identification number down at the bottom there, 734. This is a distribution of a widespread hybrid genotype. It's found in 13 lakes. Most of them are over in the East Metro, Washington, and Ramsey County. But there is 1 lake out in Wright County now that we've identified this widespread hybrid water milfoil. So all this distribution change, this is all due to clonal spread from those lakes. So it probably arose in one of these lakes. We don't know which, and maybe one we didn't identify, and then now it's been spread by either birds or boaters or combination of that to these lakes via clonal spread. Some genotypes are actually shared across the region. So across, and actually I think we'll show more information on this, but genotypes that are found in Michigan can be found in Minnesota or found in Wisconsin. And furthermore, there are these genotypes that are resistant, say, to fluoridone and 2,4-D that have been identified in Michigan. So that's sort of important. There are known fluoridone and 2,4-D resistant strains there, and we have to be worried that they could end up showing up in Minnesota. One other quick comment on genotype distribution is Lake Minnetonka. So this is the first occurrence of Eurasian water milfoil. It's large, it's got a lot of bays. If you drop the water level a little bit, it would probably be 20 individual lakes. But within these bays, there's a lot of diversity. So the bays that we've sampled have 5 to 9 genotypes each, plus tend to have either Eurasian northern present. Smith's Bay's got 9 genotypes, North Arm 8, Phelps Bay 6, and Gray's Bay 5. So there's a lot of genetic diversity now that's been generated and then moved around within Lake Minnetonka. Some of these are found in multiple bays, and there still are a few genotypes that are only found in a particular bay. Remember, that's where sexual reproduction is occurring. Well, how are we going to look at response to management? So we're concerned about potential resistance and invasibility. Katie Gannon, a student of Ryan's, assessed response to these different genotypes to treated and reference lakes in the metro before and after treatment. And based on that work, and then also some recommendations of managers, we decided to look at use these to identify potentially problematic genotypes, ones that became more frequent, ones that were widespread, to end up doing further assessment. So if you really want to know whether these genotypes are resistant to herbicides or whether they're going to be problematic, even if they're like much better growers and going to be more aggressive, you really need to do controlled laboratory experiments. So that's what we've done. What we've done now is we've exposed known genotypes to controlled herbicide doses in a common garden experiment with controls. And then from that, we can actually determine whether this genotype seems to be resistant to an herbicide or is a much faster grower. So based on our management monitoring and input from managers, we selected 6 genotypes to assess. The widespread Eurasian water genotype, so our plants came from Christmas. We also looked at the widespread, hybrid, which is from Otter Lake, and that was one of the original hybrids that Michael Moody ended up identifying. So that's found in 13 lakes. A hybrid from Ham Lake, hybrid from North Arm, a hybrid from Smith's Bay. And then based on management response, also, we looked at northern from Phelps Bay, a genotype from Phelps Bay of northern water milfoil. So for each of these populations, we went out into the field, collected individual root crowns from the target populations. We sent them sample of that to Ryan's lab for genetic identification, determined what we had. We propagated then those ones that we selected with cuttings in a greenhouse culture. So we're maintaining all these by clonal cuttings in the lab, so we know that they're all the same. For our tests, we end up planting cuttings of each genotype into cups within tanks. We end up usually using 3 cups of each genotype in a tank. We allow them to root and grow and establish for 4 weeks. And then we're going to treat those tanks with different levels of herbicide. So no herbicide, a quarter of the standard dose, a half dose, the typical dose in the treatment, and then 2 times the standard dose to look at their response. So for 2,4-D and triclopyr, these are both auxin-mimic herbicides. We ended up exposing them for 72 hours before we exchange the water. So we've got a 72-hour herbicide exposure there at the full dose. And then after we change the water, and we've got chemical analyses to back these up, we allowed them to grow for 4 weeks and look at the response of the plant. So the plants were already up to the surface. You can see here that we're growing, they're dosed, and then we end up looking at how they respond at the end of 4 weeks. We sample the shoots and roots for biomass, and we do this 4 times. So we repeat the whole experiment 4 times with 4 independent replicates. For Procellocor, another auxin-mimic, we use the dose based on whole lake treatment. Procellocor's dosing is a little strange, but these are the PDUs we have here. Probably the standard dose would be 2 PDUs for a whole lake treatment of that. And then after, in this case, 24-hour exposure, we exchange the water. And with Procellocor, we could go back and look at a much shorter exposure. That's typically what sort of thought would be important, but we were being conservative here and exposing it for a full 24-hour before we eliminated the herbicide. And then again, we allowed them to grow for 4 weeks and then sampled for biomass. And we've got 4 replicates of Procellocor also. So this is our results for 2,4-D. And now you can look, this is the Mean stem weight is a proportion of the control. So you start off with the control here at 1, and then you can see at these higher doses, you end up with a much lower amount of milfoil there. So Christmas Lake, again, our Eurasian water milfoil, you can see a rapid exponential decline here. And by this second treatment, 0.5, it's down to what, about 10% of the Eurasian water milfoil that's there. So it's really well controlled by 2,4-D. But we've identified 3 genotypes: Ham, hybrid water milfoil, Smith's Bay 299 hybrid water milfoil, and then also our Phelps northern water milfoil that aren't controlled at these lower doses. And you can also notice that for both Smith's Bay 299 and for Phelps, even at 1 milligram per liter, you end up having 50% of the plants remaining. So these look like they show some evidence of resistance to 2,4-D. The other genotypes don't have as much a response on 2,4-D. So just giving you an example, this is a control on the left. You can see there's lots of healthy plants there. This is 2 milligrams per liter. Our treatment, you see most of those plants are brown and dying. But again, I've circled here some northern water milfoil, and I think some of those in the back are northern water milfoil, and you can see that they're doing fine. So you can get an idea that this Phelps Bay seems to be resistant to 2,4-D. Most of the genotypes also grew similarly. So now this is just the actual dry weight. You can see, except for the northern water milfoil is a much slower grower. But originally we thought that perhaps resistance could be due to the plants being able to outgrow the herbicide, but it looks in general that it isn't these just being a faster grower here. Eurasian water milfoil is fairly fast growing, that isn't an explanation for the response to the herbicide. For triclopyr, much better control across all of these. We've identified a couple genotypes, Phelps perhaps, and Ham, that look like they may have a slight resistance to triclopyr, saying up to that 0.5 dose. But the other genotypes seem to be fairly well controlled with triclopyr. And with Procelicor, all of them are controlled, certainly at higher doses. At the lowest dose, we've got several Ham, Smith's Bay 299, and Phelps, that there's some question at that very low dose that they may be showing some resistance. So that's why it might be worth going back and looking at Procelicor at a much lower, shorter exposure time and see what kind of results we get at that level. So this is an example of Procelicor doing a pretty good job of controlling the plants at 4 micrograms per liter before we're ready to sample. So for our challenge tests, overall, we've got evidence that 3 of these genotypes are resistant or tolerant to 2,4-D. North arm has a bit of a shallower slope, so it may show a little bit of resistance. The lack of response to the herbicide isn't due to the outgrowing herbicide, and both the roots and stems in these experiments are similarly controlled. Notice that the widespread Eurasian genotype, and that's probably what the original target when people were working with these herbicides is well controlled by 2,4-D. We've got less evidence of resistance to triclopyr and Procelicor, no control at the lowest triclopyr dose for 2 genotypes. And for Procelicor, Ham, Smith, and Phelps show no control at the lowest dose, but the target's typically much higher at 4 micrograms per liter. Widespread Eurasian water milfoil, again, is well controlled by both of these, and we should consider shorter exposure times for Procelicor here. So we've identified several resistant genotypes, including the northern water milfoil genotype from Phelps Bay. And actually, this could be a source of resistance for some of the hybrid water milfoil. So it could be a progeny of crossing with Phelps northern water milfoil or related northern water milfoils. Several genotypes were identified for testing based on genetic monitoring. And the response of these different genotypes to oxymimics isn't the same across genotypes, right? So in other words, it isn't one that's necessarily resistant to 2,4-D is going to be one that's resistant to triclopyr. And growth doesn't seem to explain our differences here. So Ashley's going to fill us in with the next part of this talk, and she's going to look at recommendations for assessing milfoil genetics for management, and then also show you a new R-Shiny app that we can map our milfoil distribution. So Ashley.
So yeah, just kind of transitioning into from Ray's data, maybe diving a little bit more of why it's important to track strains in your lake. So obviously Ray's data did a really good job of showing that strains can be different and respond differently to herbicide treatment. There have also been plenty of other studies that have been performed that show this similar thing. Chork and Thum in 2020 did a good job of doing that, and same with Thum 2020 et al. And if we don't know anything about the strains, there are steps that we can take to prioritize that strain for characterization. And then if we do know something about the strains in your lake, it can directly help inform management. So there's two really good examples. Ray actually did touch on these examples of genotypes that we know something about and are a good baseline for, what happens when we see them in your lake. So there's that widespread Eurasian strain that Ray was talking about a lot, that E-Misgip 734, and then the North Arm strain HMYR 5016. So the E-Misgip 734 strain obviously is very widespread, not only in Minnesota, but across multiple states. It's actually found in over 100, or I think around 103 lakes. So pretty widespread. And as he mentioned, it responds pretty well to herbicide treatment. We haven't gotten any reports of any issues in treatment. And then our in-house herbicide characterizations also have shown that it is susceptible in its response to fluoridone and 2,4-D treatment. And then the 5016 strain is one of those potentially resistant strains that Ray was talking about in his presentation. It's found in multiple bays in Lake Minnetonka, but really only found in the Lake Minnetonka area. However, that could change if we increased sampling around the area. But there was some reports of some treatment issues, so we took it into the lab and characterized it for herbicide treatment. And it did turn out that it doesn't respond very well to the lower doses of 2,4-D treatment. So potentially resistant to that treatment. And then fluoridone, it responds well to, so we would say it's a susceptible strain to fluoridone treatment. So how do we prioritize strains for this herbicide characterization? Well, we can ask ourselves a series of a couple questions. The main one, has it been seen elsewhere? So has it been seen in other states? Has it been seen in other lakes? And if it has, it might be helpful to ask other lake managers who manage lakes that have also seen that strain in their lake, and maybe see how they're managing the strain or just the strains in their lake in general. And then also maybe eventually pooling resources together and creating a management plan. And then another key thing that we can look at to see if we want to prioritize a strain for characterization is to check if there has been a change in strain frequency. This can be done by doing a pre and post treatment survey. I mean, we wouldn't do this for every single lake, but it's definitely an option to think about, especially if there's kind of some question in how good the strains are responding to your treatment. And then we can also we always want to make sure that we're quantitatively monitoring for the treatment efficacy whenever possible. When we're talking about sampling, where do we start? Well, we want to start off with an initial genetic survey to get an idea of what is in your lake. So what is a representative sample? It is a random collection of individual plant stems that it's usually between 20 and 50 individual plant stems, depending on the lakeside, from across the entire lake that captures the lake-wide diversity. What it's not is going just to your end of your boat dock or end of a boat ramp and just collecting 20 different plant samples and sending them in for genetic analysis. That does a really good job of explaining all of the variation and strains right next to that boat dock or that boat ramp, but not really everything that's present in the lake. So we want to make sure we're getting everything from the lake, not just one little area. With that being said, how should those plants be collected? Well, there's many different ways that they can be collected. They can be collected through like meandering shoreline survey, a point intercept survey. We could do random stratification of milfoil beds, just kind of depending on how like your state agencies require that, or how the lake is set up, determines how the best method of sampling. So why do we want to collect a representative sample? I kind of touched on that a little bit when we were talking about what not to do. But it's really important to collect a representative sample of what's in your lake, because we want to know if we have one or multiple strains in that lake, just so we can know what to do in our next steps. So one tool that I built for us to kind of help see what's in other lakes and see what's going on everywhere else is this Milfoil Mapper app. So it shows every single strain of watermilfoil that we have ever characterized, and it's interactive. It's up to date as far as the beginning of this summer. So if you've sent in samples recently, it might not be up there. So once you open the app, you're going to have this little pop-up button, and it's going to explain how to use this map. It just kind of gives a description of the map, what's included in the rest of the website, how to use the map, links for more information, and how to bring this back up if you ever want to use it. And then we just press OK to get rid of that. And then all of these points on the map represent an individual strain. We can click on it and a little box pops up, shows all of the data associated with that individual strain. It's in Minnesota, South Lindstrom Lake. It has that lake water body ID for those of you who that is useful to. And then it also has these clickable boxes where we can get a little bit more information about our herbicide response. And this strain actually is that widespread hybrid watermilfoil strain in Minnesota that Ray was talking about previously, HMYR 4866. And we have done herbicide assays on it. And as of right now, it responds pretty well to herbicide and would be susceptible to both 2,4-D and fluoridone treatments. And instead of just clicking on all of these points, if we had a certain lake in mind, we can select a state. We're going to select Minnesota because that's probably what everybody is most familiar with here. And I'll just pick Hennepin County because that has a lot of lakes in it. And then we can also pick a lake within this state. We'll do Christmas Lake. And Christmas Lake, we can click on our little dot, pulls up the same information that we saw previously at that wider, more zoomed out scale. And it tells us that in this lake, we have the widespread 734 strain, and then we also have some northern watermilfoil. In this app, we have not kept track of northern watermilfoil strains just because they aren't managed. So sometimes it'll say just flat out northern, or it will have like NREF and then a sample number behind it. But that is all explained over in this strain nomenclature whenever you would like to play around with it. We can also clear all of our filters. And if we were interested in printing a list of lakes in a given county because you want to know, well, that was not a good example. We'll just go back to Hennepin because there's multiple lakes. We can print a list of lakes here, and it's going to give you every single lake that is in that county. You can also search by a strain. So if I wanted to search by that widespread strain, that 734 strain, now all of my markers show where 734 is found throughout the US. And I could print a list of all of those states. So if I didn't know much about that strain, which we do know quite a bit about it, it would be helpful to be able to contact other managers from those lakes. One more thing too, this is still in its first steps, version 1. So I appreciate any feedback that any of you guys have, any suggestions after playing around with it a little bit. I think maybe John put the link in the chat. So if you guys are more than welcome to play around with it, do what you will with it. And so our next steps would be to continue to genetically fingerprint and identify new strains through annual watermilfoil sample collection. And then we also really encourage more people to incorporate genetic surveying monitoring into their Eurasian watermilfoil management plans to help identify strains to prioritize for herbicide characterization, so we can put more points on the map for you. And we also just have more knowledge in general about Eurasian watermilfoil and it's how to best manage it. So just kind of some conclusions and recommendations from mine and Ray's work to continue the genetic monitoring of management responses, can help identify those problematic genotypes. The laboratory challenge tests and herbicide response assays can determine responses. Future work with genotyping by sequence may enable identification of genes or regions associated with resistance. And then the genetic composition should be considered in management decisions for lakes. And we would like to thank all of our funders. A lot of this work was funded by MASERC. So thanks to all of you who helped make this happen. And we want to thank Montana State University and U.S. Fish and Wildlife Services and the Aquatic Invasive Species Interjurisdictional Grants to the Great Lake states and tribes. And thanks for all of the advice and the assistance of numerous DNR and AIS specialists and lake consultants or lake managers and consultants. And then all of the assistance provided by Alex for his help over the summer and helping me create the website, Miller Kimball, Jane Feely, Hannah Hoff for doing a lot of the genotype herbicide assay stuff, and Greg Chorak as well for all of his additional help. And with that, I think we can probably open it up to questions, right, Alex?
Yep, that's right. We'll start with this first question that came in. I think it's a good one. Are there data available on native plant responses to the same dosages of pesticides that you guys were using in your lab experiments in order to sort of be able to compare and contrast or examine the advantages and disadvantages of trying to use a level of control on these different genotypes, given the potential for collateral damage?
That would be a big grant. Other than probably some initial work that people would have done looking at doses on that, and then trying to think about genetic diversity, right? That would be work. But that is one good point for, identifying these things, even if you need to use these. They're not real resistant, but again, a lot of times we try to manage things by going to the lowest possible level to control things. So again, even if you've got resistance and may not be resistant at a high labeled dose or something like that, but that is why it's sort of like you do want to sort of look at things that aren't responding at even some of those lower doses.
Thank you, Ray. I think we've got some like a follow-up question here of a sort. So based on the sort of herbicide trials that you folks have done so far, there was some concern or worry, at the lower levels for some of these strains. What would be sort of your pushback or counter argument to somebody looking at those data and saying, well, then we should just use really heavy doses, or we should use cocktails and just obliterate them, especially to maybe prevent them from getting any more resistant? The concern that maybe some folks will take that as an invitation maybe to ensure that they will successfully control no matter what strain might be present.
I guess I could take a quick stab at that. Ryan may end up having some input, but it's rare that you're going to sort of really eliminate all of those plants too at that really, really higher dose, and your collateral damage you have is probably going to end up being fairly significant. So with the ones that are not responding at those lower, at those really marginal doses, I think there's probably not a big concern with there, but you need to, if you go in and treat it with that herbicide and now do end up with a response, and that strain, then maybe, and you don't get a good control, then maybe you do need to go in and look at a different herbicide, right? So again, you can sort of combine that information and sort of try to pick an herbicide that's going to work for this particular strain that you're dealing with.
Thank you, guys. So the next question, I'll sort of combine a couple of questions together here because there's been a general interest. Some folks are wondering which labs are doing the testing and where do they need to send tests if they want to have the genotypes, genotyping done work, and how much will it run them and how long will it take them to get the results back?
Yeah, so lots of components to that question. Ryan's lab, the lab that I'm a part of at Montana State University, does all of the genotype testing. So you can send them to us as long as you have that link that was in the chat. The Thum lab website link is also embedded in that website. It might just be easier to Google Thum lab Montana State University. But on our lab website, we have a bunch of information for how to sample and send plants to us and where to send them. I do also recommend that you at least send us an email and kind of give a little nugget of information as who you are, what's going on with your lake, and kind of what you're looking for. And we could set up a meeting where it was me and whoever is interested in sending in samples, and we could go over how to sample the tests. There's two different tests that we can run. We can run that taxonomic ID, so it's telling either it's hybrid, Eurasian, or northern watermilfoil, or we could do another test that is telling that strain level ID. That is all shown on the website that I showed you guys. The main difference between those two tests, other than like the information that you get out the back end of them, is that the turnaround time is a little bit different. So we usually, it's a shorter turnaround time for the taxonomic test because it's a little bit easier to do. And then the strain level one takes a couple months, I would say, just because there's a lot more to do with it, and we want to try and get a nice bundle of samples to run together just to make things more efficient for processing and scoring and analysis and whatnot. The cost is $50 per individual plant sample. So an individual fragment is $50. So if you send me 2 plant fragments, it's $100 to process those. And it's the same for whichever tests that you want. And that, I'm more than happy to go over more of that in individual meetings with people who are interested in sending in plant samples.
Was fluoridone included in the lab studies? And if not, why not?
Right. Yeah. So the reason why we didn't include them in our challenge tests here is it's sort of a length of time issue. So what you saw sort of, it's 8 weeks to just do one of these oxymimic herbicides. Fluoridone, if you need to have a 60-day exposure, that's a much longer exposure time on replicating the experiment. So Ryan's lab has been set up and has done fluoridone assays with some of the strains. So it's sort of a logistics thing of what you wanted to go. So we decided to just stick with the oxymimics for our purposes.
Okay, keep your questions coming here, folks. We do have a couple more minutes left. Ray, maybe this is aimed more at you, this question. It's sort of a more general question, but it kind of speaks to that, that wonder that we all have is like, why is this special? Is there something unique or special about Eurasian watermilfoil that you think has really helped it achieve this level of success and may now also be sort of moving into the hybrid that is allowing it to succeed where so many others like it might fail?
Well, it's probably a little bit its growth form and a bit like curly leaf pondweed. It can grow in cooler water temperatures, and it has a root crown. So the next spring, those plants get a bit of jump start on some other plants and coming up. And it's clonally spread fairly easily. So that's another factor. So it can both reproduce sexually. The reason why the hybrids, and we're seeing that diversity now with them, is we are getting a lot more genetic diversity. So if you look at that original Eurasian watermilfoil, it was all clonally spreading and didn't seem like it was reproducing among itself at all. So there's very little genetic variability there. There were probably only a couple strains that were introduced originally in the 40s to North America. But now, once it started interbreeding with the northern and then it can back cross, now you end up with, just by chance, you can get things that grow better or do better in this environment or are resistant to that herbicide. So that's where now we're starting to realize that genetic diversity of the hybrid is really the thing that gives the hybrid some concern. There are some hybrids that are going to be really wimpy. There's probably a lot of hybrids that show up in a lake for a year or two and just die off and nobody ever sees them.
Great, thank you guys. I will have us end this discussion here, although I'm sure Ray and Ashley would be happy to take additional questions offline via email if you have additional. I would like you all to sort of join me in thanking Ashley and Ray for their presentation today.